Hey everybody, thank you for watching my talk. My name is Carl Hutter, and I'm a postdoc at Louisiana State University in the Museum of Natural Science. Today I would like to talk about a sequence capture probe set I developed for frogs called FrogCap. And this probe set uses a modular approach to address the challenges of capturing markers across broad phylogenetic scales. In systematics and population genetics, there are a variety of approaches that can be used to obtain genomic scale data. These methods have their own sets of advantages and different disadvantages, from differences in cost to the phylogenetic breadth that the method can work with. For example, RAD-seq is the most affordable method, but there's rapid loci drop-off such that when you sequence species outside your target genus, there may not be any orthologous loci to align, as the loci drop off very quickly from your focal group. And here for this study, I chose sequence capture as it's the most customizable and can work across broad phylogenetic scales. And I wanted to design this sequence capture probe set to work across the entire radiation of frogs, which is summarized here in this slide, with first in orange the archaeobatrachian frogs, which is about 400 species and contains famous representatives such as Xenopus and the red-bellied toads. These are the early diverging lineages. Next is the Hyloidea lineages, which is the most species-rich and contains about 3,600 species. And these are well known from representatives in the New World such as toads, glass frogs, poison dart frogs, and tree frogs. And finally, there's the Ranoidea lineages, which are mostly from the Old World and are represented by ranids, microhylids, and flying frogs and mossy frogs. And the major goal of this was to get this probe set to work across this entire 200 million year radiation. Other probe sets existed at the time when I designed this, such as UCEs, but in UCEs only about 50% of the target UCEs were captured. There are other more specialized probe sets, such as the Afrobatrachian probe set, but these only worked in smaller groups. And finally, there is the anchored probe set, but this was proprietary until recently, and also designed their markers to have a more dense tiling scheme of baits, such as fewer markers were selected for sequencing in the end. When you use a less dense tiling scheme, you can include more markers, and that's was one of the major goals of this study, was to have as many markers as possible. An important challenge with designing probe sets is the sample divergence from the probe sequence. This is especially true on large phylogenetic scales. Generally, greater than 35% of the sequence is enough to not capture that marker. And in this study, I used two main strategies to address this problem. The first was to select loci that are well represented across frogs, and the second was to create consensus sequences of the different samples that have sequence data for these different loci from transcriptomes or genomes, and then these consensus sequences, in theory, should reduce the divergence from the probe sequence and capture more effectively. From these questions, I developed FrogCap, which is a probe set for all frogs that contains a variety of data types that is contained in this large database of markers, which can then be subsetted into different modular probe designs that depend on the question. This paper describing this in more detail has been published in Molecular Ecology Resources, so you can refer to this paper for more details. And why modularity? Why is this important? This is a key feature of FrogCap, where the probe set is designed to contain individual discrete units that have different properties or applications. And this is great because these different data types can provide flexibility and variety and provide comparisons that you wouldn't normally be able to do. And some examples might include exons can be used for selection tests, UCEs for questions for neutrality is important, and comparing among different data types can be really useful if you have a difficult phylogenetic question that you can't quite figure out with a different data type. FrogCap contains 4 different types of modularity, or different types of data that can be squeezed out of the entire data set. The first type of modularity are simply just different data types, such as exons, introns, UCEs, which can be separated and analyzed separately or analyzed together. The second type is subsetting an existing probe set, where you can take the larger set of markers and reduce them down to types of markers of interest, such as sequencing only UCEs or exons, or functional loci, or selecting markers that work successfully in a specific target clade that you're interested in. And then third, the SNP data extraction. This is where SNP data can be mined from any of the target markers, with the limitation being the depth of coverage for sequencing, where lower depth you might not want to extract SNPs because there might not be a lot of support for them, but typically there's much higher depth, and here is where you would want to extract the SNP data, as it would be useful and strongly supported. And finally, the last type of modularity is mitochondrial genome extraction. While the mitochondrial genomes weren't designed explicitly with probes, they can still be extracted from the raw read data as bycatch because the mitochondria occur in high abundance within a cell, so they're sequenced incidentally, as because of the high abundance, they're not removed during the the sequence capture step. And going into these in a little more detail, for the specific data types, they all have the very different properties that can be useful for different things. UCEs, for example, are non-protein coding, have a mostly unknown function, are conserved throughout the tree of life, but they have these characteristic flanking regions that are really variable, that are useful for all types of questions. And the major advantage here is that since it's conserved throughout the tree of life, you can make phylogenies across scales that large. And second, the exons, these are protein coding, on the other hand, functional and under selection, and are moderately conserved. They also have introns, which behave similarly to the UCEs, where the introns are non-protein coding, flank the exons, and are highly variable. And these can be separated and analyzed separately, where you can take the introns and concatenate them together and have like intron alignment for a more neutrally evolving marker that for certain types of analyses. Or the exon can be used to test for selection and see if there's any patterns with functional loci that are well known in function. And for functional loci, these are markers where there's a priori knowledge on the function of the marker, and here you can test for selection or exclude from phylogenetic analyses. And the final data type is the genes, which are the exons but concatenated if they're from the same gene, assuming no linkage. And here you can get larger alignments, so you can have better species trees. And for our second type of modularity, this large database of markers can be reduced into smaller configurations based on the goal of your project. Specific data types can be selected or excluded based on length or based on how successful they were in a specific clade of interest. And additionally, we provide trial run data from which markers can be reduced based on specific clades from a well-sampled phylogeny across all frogs. And then finally, these subsets, you can use fewer probes, which can provide other advantages such as multiplexing more samples for sequencing and reducing overall costs. And third, for SNP extraction, here we can extract the SNPs from coding or non-coding regions and use them for demographic analyses or phylogenetics. And last, mitochondrial genomes, these can be extracted from the assembled contigs with great success, and they can also be annotated and aligned based on their type and can be combined with data that already exists on GenBank, increasing the modularity potential. And then returning to actually designing the markers, the major problem, again, is that too much divergence from the probe sequence will fail to capture the target. And the two strategies here are selecting the loci well represented, and here I use a genome-based strategy where I use the genome sequence from the design genome, selecting sequences conserved enough that they would be well represented across all frogs. And the alternative strategy I will compare is creating consensus sequences with alignments of divergent taxa that could provide better capture success if you have enough of the taxa from a large phylogenetic breadth that you would basically have very even divergence from the probe sequences across that entire clade. And to incorporate these main ideas and test their effect, I aim to address these main questions here. First, how does capture success vary across different data types and phylogenetic scales? Here I will compare different data types at different scales, shown here, such as extrons, introns, SNPs, and mitogenomes. Second, are the probe subsets effective? And here I will design a reduced version of one of the larger probe sets and assess capture success and depth of coverage to see if reducing and focusing your probe set on a specific clade of interest can increase capture success and depth of coverage. And finally, does a consensus-based design lead to greater capture success? Can this provide us with more data and less missing data using when compared to a genome-based design? And for the first probe set, which I call the Ranoidea version 1 probe set, is the genome-based design, and this uses the Anuran Parkeri genome as the base. And to actually assess whether these markers are well represented across frogs, I reassembled 25 frog transcriptome datasets already previously published on GenBank. And then I pairwise matched these transcriptomes to the genome and clustered matches that were together from the same gene so that I could obtain alignments and assess these alignments. And then for the Hyloidea version 1 design, which is the second probe set, I used a combination of different approaches. There were no genomes available for this clade at the time, so we had to rely on transcriptomes and the Anuran Parkeri genome. In blue, 13% of the baits and 32% of the markers are conserved enough to where I was able to use the Anuran Parkeri sequence and be within the threshold to where the probes would still capture these loci. So these are basically very poorly variable, but have enough variation where they can be captured across all frogs and provide a nice backbone. And then second, there were orthologous loci shared between Hyloidea and Ranoidea, and the problem here was that the markers were too divergent, so they wouldn't be captured in Hyloidea. So here I took the transcriptome sequence data for these markers and created consensus sequences within Hyloidea while maintaining the orthology with the Anuran Parkeri genome so that these data can be combined into a large Anura dataset. And then finally in green, the unique transcriptome, of which about a third of the Hyloidea data is represented by. These were completely new markers only found in the transcriptomes that were also created with consensus sequences. And then the final probe set was the subset design, which we called Reduced-Ranoidea, which was used the half-size probe set of 20K probes versus 40K, which the other two had. And here I selected markers that were successfully sequenced from the trial run for the project target taxa Psittazyga. And here we wanted to know if the depth of coverage increased and if multiplexing more samples could be beneficial. And then with our final set of markers, we filtered them all after gathering statistics from the alignments to ensure that they all had greater than 5% parsimony informative sites. And we also wanted to maximize phylogenetic representation, so we made sure that 80% or greater of the samples were found in the alignments for a specific marker. We also filtered for duplicate matches and paralogs. And finally, we used 120 base pair length baits across the length of the marker, tiling 2x, with a 20 base pair intron buffer but this intron buffer was only applied in the Ranoidea version 1 because this was the only one where we had a genome and intron sequence could be used. And then to summarize these final probe sets, I have all three listed here, with the Ranoidea set on the top, designed from the Nanorana Parkeri genome, the Hyloidea and Archaeobatrachian set, which is the transcriptome consensus sequences, and then finally the Reduced-Ranoidea set, which is designed from the full Ranoidea set, but reduced down to a smaller size to fit within the target clade of interest. And to summarize all the probe sets sort of together in their different data categories and marker size classes, I have these two figures shown here. And combined on the very bottom is the full FrogCap marker database, of which these other three probe sets are derived from. And then the other three are the probe sets used in this study. And for the first data type, the legacy data type in purple or pink, these are hard to see because there's only about 80 of them in each of the data sets. UCEs, there's about 2,000 in each, and exons is the most abundant, being represented by several thousand for each of the probe sets. And of these exons, they are found in different size classes shown on marker size on the right, where the majority of them tend to be small for mining SNP data. However, there are quite a large number of large alignments greater than 1,000 base pairs, where we have about 500 in each of the probe sets. And to demonstrate how these probe sets overlap and relate to each other, I used a Venn diagram to provide this information. The dotted lines represent an updated version of the probe sets that won't be discussed much here and is discussed more in the paper, but they exclude markers that did not work in the trial sequencing run and include some new legacy markers. Otherwise, in green, Hyloidea version 1 has 3,099 unique markers and 6,130 that overlap with Ranoidea version 1. Ranoidea version 1 then has 7,387 unique markers. And then Reduced Ranoidea has 3,247 markers with partially overlaps with both the data sets. And once the probe sets were designed, we wanted to test these, and we included a test set of 121 samples from several different phylogenetic scales. The order scale, which is the Anuran level, contains the 24 samples from the two superfamily scales, so Hyloidea and Ranoidea, and here this scale only contains the orthologous markers between the two, which is about 6,000. And then we included the family scale of Mantellidae, which includes one species for every genus, and the genus scale, which we use the genus Cornifer. Here, this included samples broadly represented through the genus. And finally, the species scale contains 16 samples from a single species from 4 different populations. So this scale will be useful for assessing variability at the population level. And for our extractions, we used a magnetic bead protocol and sent the samples to microarray, or now called Arbor Biosciences, for library preparation. They used Illumina TrueSeq libraries and hybridization-based enrichment, where we multiplexed 8 samples per pool and sequenced on Illumina HiSeq 3000 with 150 base pair paired end reads. To analyze the sequence capture data, I created an R package called PhyloCap. And this R package can process your data all the way from raw reads to alignments and trees. And this is a more easy to use alternative to PhyloCap, which is only for UCEs, and it can work with any sequence capture dataset. And essentially, like every other pipeline, it's a wrapper for a bunch of different programs with custom functions and code to clean up and move files around and keep everything nice and organized, and to greatly simplify and streamline the process. This can also easily be installed via Anaconda and is available on GitHub at this link here. The R package first pre-processes raw reads by trimming them of barcodes and adapter contamination using the program FastP. And additionally, the program removes contamination from outside sources such as bacteria, Drosophila, humans, and other common organisms. And this works by matching the raw reads to genomes from these organisms and removing them if they have a 100% match. And after cleaning the reads, the paired end reads are merged together using FastP. Exact duplicates are then removed and the merged reads are assembled using spades under several different Kmer values. And once we had the assembled contigs, we matched these contigs to the probe sequences to identify and annotate each of the contigs to see which marker it belonged to. And here we detected and removed paralogs and merged any markers that were discontinuous. And finally, we aligned all the samples from the same marker using MAFT and separated the loci into the 4 categories of exon, genes, introns, and UCEs. And the alignments were finally trimmed where each data type was trimmed a little differently. The exons were trimmed to maintain an open reading frame, while internal trimming was applied to UCEs and introns. External trimming was applied to all alignments, where the edges were trimmed to make sure that at least 50% of the samples had sequence data at these ends. And these trimmed alignments are ready for downstream analyses. And to mine for the SNPs, the pipeline forks off at the clean read stage, where the cleaned reads are mapped to the assembled sample contigs using BWA. These mappings are further processed using SAMTools Picard and GATK4 following the best practices pipeline. And these sample contigs is basically a reference, are used as the reference for the mapping, and each contig has their variants identified and filtered using Phred scaled quality scores, where a score of 20 was considered strongly supported. Then returning to these questions, the first was, how does capture success vary across different data types and phylogenetic scales? When comparing across phylogenetic scales, which is shown here on the x-axis for these bar plots, we find some variation among scales in a few important ways. In A, the number of alignments, at more shallow phylogenetic scales, like the family level and below, there is substantially more intron sequence. This is likely because there's too much variation in the introns, so at broader phylogenetic scales, the intron is very difficult to align and is thus trimmed out by the trimming programs. While at more shallow scales, from family and genus, the introns are more similar to each other and can be more easily aligned. And this is also reflected in the number of base pairs of intron, again shown in green in B, where the family level at Mantellidae has a lot more bases of intron than the other scales. And when assessing variability at different phylogenetic scales, we find that broader scales have much more variation in all data types. So the Anura and superfamily levels, everything is super variable. At the shallower scales, such as family and within a species, while the variation isn't as high, there's still good variation here, especially in the introns. So this probe set has been shown to be useful for species delimitation and gene flow, as there is enough variation in the exons and introns to be useful for population genetic analyses. And then to display this in a phylogenetic context, here's a tree of the 48 Anura dataset from the orthologous markers in that dataset. And the x-axis by the bars is the proportion captured, and you can see how much variation in success there is most in blue had a 75% or greater proportion of samples captured, and only a few were a bit lower, which I'll explain in more detail later. For extracting SNPs, when assessing our ability to do this with our marker set, we find great success as well, where on the plot on the left, that shows the initial pool of SNPs that we found without any filtering. When aggressively filtered, the second plot, which includes only high-quality SNPs with a quality score greater than 20, we still find thousands of SNPs, which nearly every marker has at least one strongly supported SNP, so that for each of the probe sets you can have up to 10,000 SNPs that are independent, or tens of thousands of SNPs if independence is not important. And extracting mitochondrial genomes was also very successful. Using the 48 sample Anuran dataset, the percent completeness for each marker for each sample is shown in the grid cells below. The majority of the markers have 90% or greater of the target mitochondrial marker present, and for markers shared in common, it was mostly successful. For example, ND5 and ND6 was the least successful marker, but this does not matter much as it wasn't shared among many samples. And the great thing about this is that now the sequence captured dataset could be combined with 16S or 12S datasets off of GenBank, which expands the utility of this modular probe set design. And then our second question, are probe set subsets effective? When assessing Reduced-Ranoidea, when we assess the capture success, we find low variation in capture success, which is great because most samples captured 95% or greater of the target markers, except for one shown here. And this also carries over to the alignments shown on the right in green, where the trimmed and untrimmed alignments had a similar amount of base pairs. So this suggests that the plan was successful, where choosing the markers that work really well in a group ends up with a very high capture success. Importantly, we wanted to address whether the reduced design led to an increase in depth of coverage for the target loci, which could be used for more confident variant calls in population genetics studies. The top row of figures has the probe set on the x-axis, Hyloidea, Ranoidea, Reduced-Ranoidea, and the normalized depth on the y-axis. The depth was normalized using RPKM, which scales for each sample scales the amount of sequencing depth so that the samples can be compared to each other. And when these samples are rescaled, we find that the Reduced-Ranoidea has about 2 or higher depth than the other samples. And and in C, the exons and introns are compared, where the exons generally have a greater depth than the introns, likely because the exons are actually targeted and the introns are incidental capture. In the bottom row, depth is plotted across the length of the contig on the x-axis with the medium depth on the y, showing overall higher depth for each bin in Reduced-Ranoidea, which looks like the video is covering, but it's higher. And C, our final question, does the consensus-based design lead to greater capture success? To assess capture success, we first measured sensitivity, which is defined as the percent of bases from target markers covered by post-assembly contigs. In other words, is the percentage of target markers captured for each sample. Next, we calculated the genetic distance for each sample as the mean across all samples. And on the left figure is Hyloidea version 1, which was a consensus design, and on the right is the genome-based Ranoidea version 1. And we find a significant and strong relationship between genetic distance on the x-axis and sample sensitivity on the y-axis in the Ranoidea genome-based design. This suggests that markers are more likely to be captured when they are more genetically similar to the genome design probe sequence. And more importantly, the results show that the consensus-based design, Hyloidea version 1 on the left, had no relationship, suggesting capture success is explained by another variable or is stochastic, but it is not explained by genetic distance. Secondly, we assessed capture success using specificity, which is defined as the percentage of cleaned reads that can be mapped back to the target markers. Higher numbers of mapped reads suggest that the target probes were specific enough to capture only the targets and not other loci accidentally, and that they also are capturing loci instead of nothing. And again, the consensus-based Hyloidea version 1 is shown on the left in green, and genome-based Ranoidea version 1 on the right, with genetic distance on the x-axis and sample specificity on the y-axis. And here in Ranoidea version 1, we show a strong negative relationship between genetic distance and sample specificity, which again shows that capture success is influenced by genetic distance when using this genome-based design. However, the consensus-based design, Hyloidea version 1 in green, there was no relationship, suggesting that the percent of mapped reads to the target markers has nothing to do with genetic distance, and that this variation is explained by something else. And this is great. This is exactly what we anticipated and wanted from a consensus-based design, that we wanted the marker drop-off to be random and not influenced by genetic distance. And then finally, the third measure of capture success is the amount of missing data. So the consensus-based Hyloidea on the left, again, has no relationship with genetic distance on the x-axis and missing data on the y. This result suggests that consensus-based design reduces missing data to a stochastic amount that is similar among species, hence the horizontal line. And this similarity would mean that some other stochastic factor is leading to this missing data, and it has nothing to do with the genetic distance. Ranoidea, which is the genome-based design on the right in blue, has a positive, strong relationship with genetic distance, where the more genetically distant you are from the target marker, the more missing data you will have. And when mapping this out on the phylogeny, we can sort of see these patterns visually. So the red arrow here is pointing to the family that contains the nanorun genome used in the genome-based design. And with increased phylogenetic distance, it's clear there's a loci drop-off effect, which goes all the way up to microhylidae in the middle there with the purple and green, with the loci dropping off from the genetic distance. And now, on the other hand, in the red box is the group of samples that was used in the consensus-based design. So here we had transcriptome sequence that we could use and create consensus sequences from. And for all these samples, the capture success was very high and uniform. And loci drop-off was only observed outside the red box, where we didn't have transcriptomes to create a consensus sequence from. When assessing different data types and the conclusions we can draw from them, the informativeness via parsimony informative sites varies, with the intron having the most variation, exons having a moderate amount of variation, and UCEs not being far behind. Different phylogenetic scales, we find that introns had the most useful variation at shallow scales because the introns could be aligned together. But also at these shallow scales, the exons and UCEs were not variable, so the probe set provides a way to have variation even at the species level. And then at deep scales, exons UCEs were more variable and thus more useful at these broad phylogenetic scale. So the probe set was a major success in being able to capture broadly across all frogs from the entire order level down to the population level. And this is supported by some published studies that have used FrogCap, with the first comparing data types, exons, introns, and UCEs phylogenetically, and that we show in this study that different relationships are provided based on the data type. So carefully examining and using different data types can be important when disentangling difficult relationships. Second, when using the probe set, using SNPs, we used and tested for gene flow and did population demographic analyses in Chan et al. 2021, and we found that FrogCap was successfully used to assess gene flow in a cryptic species and determine the species status based on the fine scale SNP data we were able to obtain from across populations. And hopefully the study has convinced you that modularity is important and that different marker types can provide utility that depends on phylogenetic scale or the question being asked. Our results show that the subset probe set had greater capture success and higher depth of coverage, so this could be a strategy to reduce costs and sequence more samples. SNP mining can also provide high-quality data for demographic analysis. And then finally, the mitochondrial genomes and legacy markers can be extracted and used alongside previously published data from GenBank, for example, to expand the modularity to previous data sets. And finally, when assessing genome versus consensus-based probe design, we find that the consensus-based design are superior and do not drop off from a predictable loci effect when comparing genetic distance and various measures of capture success. The genome-based designs all have significant relationships between capture success and genetic distance, which shows how using a single species genome sequence can capture success and how a consensus-based design can lead to more even and broad ability to capture samples across large phylogenetic scales. And all the data and the probe set and everything in the study is freely available for any researcher to use or modify how they like, as I am very committed to open science and making everything transparent and reproducible. All the pipelines used for all data analysis are available on GitHub. The raw read data is available on the NCBI, and the contigs, alignments, and transcriptomes are all available on the Open Science Framework. And thank you for listening to my talk. If you are interested in the probe set or have any questions, my contact information is on the bottom below. And I would like to thank my PhD advisors, Rafe Brown and Rich Glore, the various people that have provided tissues for this study, and all the funding sources shown here for making this study possible. Thank you.